you for inviting me to speak at the Prevent Cancer Foundation Quantitative Imaging Workshop of this year. I've been asked to talk about metrology and quality assurance perspective of what are the challenges and hurdles hindering the validation and regulatory approval of AI algorithms for CT screening. Here I list my findings and conflict of interest of grants and others, as well as the University of Chicago policy. Now, to consider the challenges and hurdles, we first need to understand metrology and QA. Metrology is the scientific study of measurement. We will need to consider performance metrics of the AI algorithm by itself, as well as the AI algorithm when it's being used by radiologists. That is, what is the performance metrics of evaluating the radiologists when they are using the AI algorithm? One needs to look at validation methods, and all of these are to appropriately get through regulatory approval or clearance. QA, quality assurance, is the maintenance of a desired level of quality in both the input data, the images, and in the AI algorithm. Performance of the AI algorithm across different populations, different medical institutions, different image acquisitions, and so on needs to be considered. And we also need to consider quality assurance as we continue performance evaluation post-regulatory approval or clearance. So for this sessions, the discussion objectives given to me are, what are the major challenges for a developer trying to bring an AI product to commercialization? How important is rigorous QA for that input data? And what will be the APM's role in MIDRIC? So let's consider medical image interpretation. Medical images are meaningless grayscale color scale patterns unless viewed and analyzed by an intelligent observer. That could be the radiologist, the computer, or a combination of the human and the computer. That is AI aided. The task of the human eye brain systems are finding and localizing a signal in an image, characterizing, classifying, or diagnosing the signal as disease or non-disease, and also clinical decision-making on patient management through integrated diagnostics and monitoring. The task of AI is very similar. Basically, we're converting images to quantitative values. So very similar to the human eye brain system, and very important to know what is the clinical task when developing an AI algorithm. And this is important to note. The AI algorithm, shown here, is basically the computer algorithm that exists in a system to take the images, process them to answer some biomedical question. How they are used is also important. For example, computer-aided diagnosis is one such method. We need to consider, will the AI be used as an aid to the radiologist? Will it be also used as an aid or as a concurrent reader? Could it be used as a primary reader in triaging, or will it be totally autonomous AI? We'll talk more about that later. And note, this still does not specify whether the AI is to help interpret an image or to also integrate it with other diagnostics. So what are the major challenges for a developer trying to bring an AI product to commercialization? Well, going from the idea to the lab investigation, proof of concept, maybe translation, getting an FDA product, installing that product, and getting it to actual widespread clinical use is a long path. Many papers out there are at this stage. It's a laboratory investigation done on perhaps an independent data set. Some might get translated, and then the rigorous of going through the FDA is quite strong and quite beneficial because that demands many of the aspects that I will mention here. The developer needs to understand the clinical task. Are you looking to detect or diagnose? Are you looking at response to therapy and using multiple images over time? Are you going to be using AI as an additive component in the decision making or as a replacement in the decision making? And are you developing the AI to improve the performance of the interpretation task, that is the efficacy, or to improve interpretation times, for example, and that would be efficiency? You also need to understand how the AI algorithm will be used in the clinical task as a second reader or a concurrent reader. Will it be used as a triage or to rule out normal cases? Or will it be used as an autonomous reader? These are questions that you need to answer prior to the development of the AI algorithm, and definitely prior to the evaluation of it. In developing, you need enough data. Data could be the images, the clinical data, as well as any annotation of relevant disease or tumors and non-disease region. You need to consider the number of cases, and not just the number, but the distribution of cases, the disease type, the severity, acquisition protocols, as well as various populations. You also need to ask yourself, who will ultimately use the AI algorithm? Will it be radiologists or will it be primary care physicians? Will they be in academic, private practice, both? And what range of training of the ultimate end user will be necessary? And then after that, you have to come up with an appropriate performance metric for the clinical tasks, such as ROC analysis, sensitivity, specificity, and others. All of this will impact both training and testing. Now, once you have FDA approval and clearance, there are still challenges to bring your AI product to market. You will have to integrate your AI algorithm into a clinical IT backbone or cloud. You have to look at disaster recovery, HIPAA IT security issues. And a big issue that I think many do not think about is the potential for the end user, the clinicians, to use the system off-label. It might be intentionally. That is, maybe it's an FDA cleared second reader AI system that they are going to use as a first reader. Not allowed, but might happen. Or it might be used unintentionally. That is, it was approved for a specific imaging protocol. They have an image, similar but different protocol, and they input it and the system doesn't work. What happens? And I think it's very important to remind the end users garbage in, garbage out. We use garbage in, garbage out when we're trying to have people train appropriately and test appropriately, but also that needs to be when used appropriately. And these challenges are not new. Let's look at what's changed with AI over the decades. We have faster computers, larger data sets, more advanced algorithms, including deep learning. We have the realization of additional reasons and means to incorporate in clinical practice, and it's now being developed for more and more clinical questions. However, the clinical task of detection, diagnosis, response assessment, those broad ones are the same. We have the same concern for garbage in, garbage out, the same potential for misuse, same methods of statistical evaluation, same need for a sufficient number of cases to span the distribution of disease and normal presentation, and same need for imaging domain experts and computer domain experts to work together. Second objective of the session is how important is rigorous QA for the input data? That is, how, or I should say, can big data overcome the issue of input data heterogeneity? Well, one way is, do we train with heterogeneous data? And we also have to think about diagnostic quality and good image quality. When radiologists read, they may get an image. It may not be the best image quality, but they will say, I can still make my diagnostic decision on this image. So it is of sufficient diagnostic quality, but may not have the best physical image quality. Do we train our algorithms on AI for that? We definitely will need to test them on those. So we can ask ourselves, should we train with bad data? Should we train with bad data to mimic clinical environment? So when we look at databases and quality assurance, we can look at the differences in image acquisition or population. If we look at different image acquisitions, we have to consider different spatial resolution, noise, and system gain. Now, out there we have big data, which is standard of care, which will be very variable. We have data from clinical trials, medium size, and we have small data. And often with AI development, people are developing with a very focused small data set that might be very carefully obtained, but ultimately it's going to have to be used on standard of care images. Population may affect the computer extracted phenotypes and the AI algorithm, such as disease prevalence, geographical variations, single versus multiple institutions, and lack of harmonization of the data. So this is so important, I'm going to repeat it again. The size and distribution of the data set will depend on the task. So if you have a very easy task, you won't need that many cases. And is it a binary yes or no type decision, or does it span multiple severities of the disease? That will also influence the size and distribution of the data set. And also the quality of the annotation and truth. If you get your data from multiple places, maybe one institution is very careful about reading that pathology report and the other one isn't. So it's always good to double check the truth. Here's an example that Whitney looked at in attempting to harmonize MRI radiomic features across two data sets, one from the US and one from China. She used the combat data harmonization feature. And if we look at this, these are cancers where the cancers from the US are in red and in China in orange. And this is one particular characteristic that one can pull from the image. And you see after harmonization, the distribution of the two features can come together. This was also shown with benign lesion, that doing harmonization was able to bring the two distributions more harmonized. But how does this affect performance? Well, she showed that if you do this and then retrain, the pre-harmonization to the post-harmonization improves the classification ability. Now, this was in malignant and benign breast images, but it shows that you can do some harmonization of features. And in this case, potentially, would you have one algorithm for both institutions? Well, she's still checking on that because perhaps harmonization might not work. And you might need AI tuned to specific populations, specific institutions, specific acquisition systems. I think when it comes to this heterogeneity and trying to harmonize a batch effect, you can think of the batches as this batch came from one institution, this batch came from another institution, this batch came from maybe the same institution, but a different CT scanner. And of course, as mentioned earlier in the day, recall the discussions on Cuba. And third, I was asked to comment on the APM's role regarding QA of data in MIDRC. So MIDRC, as you might have heard, is a new NIBIB funded initiative. It is called the Medical Imaging and Data Resource Center. I am the PI of it, and is hosted at University of Chicago. It is co-led by APM, RSNA, and ACR. And this is because of the COVID-19 pandemic. We need to develop new methods for surveillance and early detection, for triaging and differential diagnosis of COVID-19, as well as prognosis, including prediction and monitoring of response. So this effort is for the rapid and flexible collection, AI research, and dissemination of imaging and associated data. Two major components of it is to create the open discovery data repository, including creation, testing, quality assurance, and data connectivity, and machine intelligent computational capabilities, clinical relevant algorithms, and software tools. This is the flow chart of it. Data intake will be between ACR and RSNA going up into a data commons, which will then be shared with users. The APM's effort is on developing and implementing quality assurance and evaluation procedures for usage across MIDRC. You see here across is the APM Technology Assessment Guidelines. This is for image acquisition and protocol harmonization. There are two major parts of it, is development of digital and physical imaging phantoms for COVID data and assessment of image quality on ingestion into the MIDRC. And there's also the APM's role in technology assessment, metrology, benchmarking, and task-based distribution for evaluation in order to expedite the evaluation of developed AI algorithms to the public. The development of benchmarking methods for the various technology assessment and clinical tasks in COVID-19 research and translation are needed and will be investigated, and the development of task-based distribution methods. Both of these are being developed to facilitate FDA clearance and accelerate clinical usage. Methods of selecting clinical cases for independent testing are required to appropriately evaluate a validated algorithm. And for specific tasks, for example, diagnosis of COVID-19 from other presentations of pneumonia, a distribution of cases matched to a clinical claim and intended patient population will be randomly drawn from a larger sequestered data set within MIDRC and subsequently used in the testing. So we've covered these discussion objectives, and I look forward to the various input from the panelists. In summary, the ways to use AI in clinical interpretation is expanding every day. We need to keep in mind that we need to evaluate it alone in the particular task, and also we need to evaluate the end user. We need to look over the robustness over the range of data quality, make sure it's being used as intended. High-performing AI used for the wrong clinical task or incorrectly by the user will not yield expected clinical outcomes. And we need to realize that AI output is not always correct. And so we need to ask, what level is acceptable for clinical use? How will it vary with the task? What is the cost benefit? And what level is high enough so explainability is not needed? I'd like to thank my lab of wonderful graduate students and research lab staff and collaborators. It's a wonderful collaborative experience. Thank you.